Welcome back. We're going to continue studying the eye here. We're going to get into how vision works. So visible light exists between about 400 and 700 nanometers. So when we look at the scale here, it's a very small piece that is actually going to make up visual light here. The colors we see is whatever wavelength of light that the object cannot absorb. So whatever reflects back at us is what it can't absorb. So most leaves are green. So what that means is that they absorb the blue and the red light and they reflect the green light back. Okay. All right, so let's talk about refraction. Light is going to travel in very straight lines. They're called light rays. But when they go through something, they tend to bend, which is called refraction. Okay, so if they come at something at an angle, they can bend, or if they come at something that is curved. So we have a series of convex structures in the eye. We've got the cornea, we've got the lens, and then we've got the posterior lens, all of which have the ability to bend light so that the light that comes in is all going to converge at a single point of contact on the retina. That's how you get clear, sharp vision. If you have multiple parts of the retina being activated by light, then you're going to perceive whatever you're seeing as very blurry. You need everything to pinpoint down to one particular spot, and that's called the focal point. That's what we want. We want one single focal point, not multiple focal points. Now, as we do this, because of the way that the light is going to bend, the image that is actually formed on the retina is upside down and reversed left to right. So here you can kind of see this concept of how light travels. So here, this is the orange point of light and this is the purple point of light, but you can see that as it goes through a lens, orange is going to end up down here and purple is going to end up down here. And then here you can see the reverse aspect as well. So orange was over in this corner and over here it's over here. So it's not only upside down, but it's also reversed left to right. That's why we use so much of our brain to process vision. We have to turn all of this stuff right side up and flip it left to right in terms of the perception. So we've got several pieces that light has to go through. So we're going to go through the cornea, into the aqueous humor, through the lens, into the vitreous humor, and eventually focus onto the retina. So it's going to be refracted 3 different times possibly. Cornea, going into the lens, and leaving the lens. Now, the lens is what will change that will allow us to have a focal point on the retina, whether the information is coming from very close up or very far away. This is where we get the concept of visual accommodation. This is the lens either thickening or thinning to allow the focal point to hit the retina. So when we talk about distant vision, so we're talking about looking at something that is 6 meters away or farther. This is what's called an emmetropic eye. Everything is relaxed. We can stare at a mountain far away from us all day. Our eyes are not going to get tired. The ciliary muscles are relaxed, okay? And that means that they're in their lengthened state. So just picture biceps brachii. When it's relaxed, it's going to be longer than when it's contracted. Because of the way the ciliary muscles and the suspensory ligaments are arranged, when the ciliary muscle relaxes, the suspensory ligaments are going to pull on the lens. That's going to mean that the lens is very thin at that point. Now, close vision is where we have a serious number of active adjustments. This is why you can't read or stare at your phone forever. So first of all, we have to accommodate the lens. The ciliary muscles have to contract. They're going to shorten. It's going to cause the suspensory ligaments to slacken up, and the lens is going to get thicker. So we've got ciliary muscles contracting. We're going to have to go through the pupillary reflex, which is going to cause sphincter pupillae to contract the pupil. And we have to go through convergence, where both of our medial rectus muscles will contract to bring our focus inward. So there's 3 different muscle groups that have to contract for close vision. This is why your eyes get tired when you're doing something close up, and you have to occasionally look up and let everything relax, let everything go emmetropic again. So let's take a look at these. So here, emmetropia is perfectly straight eyes staring off in the distance. Beautiful little farmhouse here, or barn, whatever that is. And we didn't have to contract our medial rectus muscles. But if we're reading a book, we have to converge. So medial rectus, possibly inferior, depending on where we're holding the book, they all have to bring our eyes inward so we can focus here. So we need to take our 2 eyes and converge on a single point out here. So that's one of our muscles. The sphincter pupillae is going to contract, and that's going to allow us, again, to narrow out the field here so that we can get a single point of focus. So we've got rectus muscles, we've got sphincter pupillae, and then finally we've got the ciliary body and its effect on the lens. In distant vision, what happens is the ciliary body, so remember that this is 3-dimensional. It comes all the way around the lens. We're looking at it as if it's only above and below, but in reality, it goes all the way around. Remember that picture we saw with the suspensory ligaments coming all the way around? They attach to the ciliary muscles all the way around. So when this relaxes, it lengthens posteriorly. It's going to lengthen and pull the suspensory ligaments taut, which is going to thin out our lens significantly here. When we're doing that, everything's relaxed. So these are nice relaxed muscles. The relaxed muscles pull the suspensory ligaments taut, and the lens is pulled thin. If I need to accommodate for near vision, the ciliary muscle has to contract. It's going to contract anteriorly. So it's going to get shorter by pushing anteriorly. That's going to cause the tension in the suspensory ligaments to be removed, and the lens is going to bulge. It's going to thicken up because it's no longer being pulled taut. So 3 different muscles that have to contract in order for this to function in terms of the near vision. So near vision is a very active process, whereas distant vision is a completely relaxed eye. Now, we definitely have some issues with refraction in terms of the shape of our eyeball, because that definitely plays into whether or not our focal plane hits the retina or not. So a couple different things. Astigmatism. This happens when the shape of the eyeball, particularly the cornea, is not perfectly rounded. And so you end up basically having multiple points of focus, but usually within a single line. Okay, so this happens when your cornea is not shaped properly. Presbyopia is the ability to loss of ability to accommodate with age. This is because the the lens tends to age poorly. We talked about that. Hyperopia and myopia are probably your most common here. So hyperopia, okay, is what we would call somebody who is farsighted, meaning that they can see far away, but they cannot see close up. The problem with this is the eyeball is too short. And so the focal plane is actually back here behind the eye. So when it comes in, when this light hits the retina, it's not pinpointed. There's multiple points of focus. We correct this using convex lenses that are going to cause everything to converge sooner. And now we have a single pinpointed focus on the retina. The opposite is myopia, or nearsightedness, where you can see near, but you cannot see far away. This is because the eyeball is too long. So the focal plane is right here. And so light comes in, it pinpoints right here, and then the thing is the light just keeps going. And so it's gonna diverge back out, which is gonna cause multiple points of focus on the retina. We're going to correct this with a concave lens that's going to diverge the light out and then allow it to converge sooner. So very common. Most people's eyeballs are not shaped perfectly, but we are usually able to correct people's vision fairly good. All right, so let's talk about how light is going to interact in the eye. Light is going to come in this direction, and the signal is then going to proceed in this direction. So light comes in here, activates our photoreceptors, which are rods and cones, and then they synapse with the bipolar neurons, and the bipolar neurons then synapse with the ganglion cells, and the axons coming off of those are going to be your optic nerve. So light has to travel past all of those to get to your photoreceptors. And then this is the pigmented epithelium back here, which will grab any scattered light, because again, we want pinpointed focus on the retina there. So that is the actual pathway of the signal transduction. It goes photoreceptors, bipolar neurons, ganglion cells. We also have a couple other cells that are going to help modify the signal. So we've got horizontal cells and amacrine cells that are going to help modify, but they are not necessarily part of signal transduction. Let's talk about the rods and the cones here. So here you can see a SEM shot of them, which is really kind of cool. And here you can kind of view the fact that they are, in fact, different. So let's talk about their functional differences. So rods are going to give us our non-color, so our grayscale vision. So there's only one visual pigment, and it's dealing with grayscale. Cones give us color vision. So we have three different pigments we'll talk about. So we have red, green, and blue. And so rods are responsible for grayscale, cones are responsible for color vision. Rods are incredibly sensitive to light, meaning that they don't need a lot of light to function. So there doesn't have to be a huge amount of light in order for your rods to fire, whereas cones have very low sensitivity and they require huge amounts of light to activate. So rods give us our vision in low light situations, whereas cones are going to function when we have bright light. Rods have low acuity. So acuity is how sharp the vision is. And we're going to see why. Okay, so we'll talk about the dual vision system and see exactly why rods have low acuity versus cones having very high acuity, so very, very sharp vision. Rods are by far more numerous. You've got way more rods than you have cones, and they're located mostly in the peripheral retina. Cones, not nearly as many of them, and they are clustered in the central retina, most of them in that fovea centralis. So if we think about this, Walk outside, middle of the day, that's cones. You're seeing in color, everything is very bright, you've got clear, crisp vision. You wake up in the middle of the night, you open your eyes in your fairly dark bedroom. You're not getting clear, crisp vision. You're not seeing the colors that are there, but you can get a decent idea for, where your bed is versus where your dresser is versus where the door is. So you're getting a fuzzy, kind of a fuzzy-ish image of what's going on there. The two of them together allow us to function within the 24-hour light cycle. If we only had one or the other, then we would be really out of luck in the opposite light situation. So let's talk about that acuity for a second. This is what's called the dual visual system. Rods do not have a lot of acuity. They don't provide super sharp, clear vision. And there is a direct anatomical, we're going right back to structure versus function here. We look at 1 millimeter squared of the retina. There are a ton of rods there. And, here we're looking at 5 of these rods converge their signal onto 1 bipolar neuron. So you have 3 bipolar neurons within this 1 millimeter space, and they all converge on a single ganglion cell. So this means that the signal that is heading to the brain on this particular fiber is covering a full millimeter of the retina, and it's not going to be super clear. I mean, you've got 15 inputs that are all going on one piece. Over here, we look at the cones. A single cone is going to take up an area of 2 micrometers squared. Okay, and every single cone has its own bipolar neuron and its own ganglion. So this nerve fiber right here is only carrying information from 2 micrometers squared of the retina versus this one that's covering a millimeter squared. So this is going to be very clear input. This vision is going to be very, very good, very super sharp because it's just this one teeny part. This is going to be less so because we've got 15 different inputs trying to converge onto this one fiber. But again, so you may be looking at this going, we'll just get rid of these. I mean, they don't seem that good. But remember that these are more sensitive to light than these. These require a lot of light to activate, whereas these don't require very much light to activate. So it all goes back to the light-dark cycle there. All right, so let's talk color vision for a minute here. What we have here for color vision is basically 3 different types of cones. We have what are called short wavelength cones that deal over here kind of in the blues, purples, blues, maybe a little bit into the greens. We have medium wavelength cones that deal with the kind of like sort of blue to green to yellowish starting on orange. And then we have long wavelength cones that are going to deal with your basically kind of from the light blues across over to mostly your reds. And then here's your rods. They lie right there. So the way we perceive different colors, because obviously we see more than blue, red, and green. Okay, we have more precise colors than that. We've got this blue-green in here, this area right here. That's not a blue or a green or a red. So the way we perceive it is the percentage of each type of cone that responds. So for violet down here, way down here, you're going to get about 50% of your short cones responding and 0% of your medium and long. So that's solely the short, and it's about 50% of them. Now, if I go to something like blue-green, okay, so somewhere like right in here, that's going to give us about 20% of short, 82% of medium, and about 60% of long. That's how we are able to distinguish these colors. Red, way over here at close to 700 nanometers, is only 5% of your long cones. So how we perceive color is based on how many of each of the short and medium and long cones are responding. Last thing is the visual pathway. So we have a pretty decent visual field here. It's not quite panoramic, but decent, okay? And when we look at how the visual fields of each eye work, so the right eye is the yellow. So the right eye basically can see all the way over here, but also all the way over into this left field. And then the left eye can see way over here, but also it crosses over into the right here. And so this is the area where both eyes can see. If we look at how the fibers travel, each eye has a medial and a lateral half of the visual field. The medial halves, okay, they actually cross to the opposite side. So left is purple, right is red. So if I follow these purple fibers from the left eye on the medial side, so these two right here, they're going to come in. Here's our optic chiasm. You guys saw that on the brain. And then they actually desiccate. They cross over to the contralateral side. And so this information is going to end up on the right side of the brain, along with the lateral from the right eye. So the lateral fibers stay ipsilateral. This is because the left brain controls the muscles on the right side of the body, and then the opposite is also true. So we need to see what's going on the opposite side of the body. So the medial half of the visual fields are going to desiccate or cross over to the contralateral side, while the lateral sides will stay ipsilateral, providing us with close, not, we're not at 180 or anything, but we do have a fairly decent visual field for the placement of our eyes. So our eyes are straight ahead as opposed to kind of out to the sides, like most animals. And so for that, we have a pretty decent field of view. And we have information from both of our visual fields coming into each side of our brain.